In health news tonight, scientists in BC announced today on World Water Day that they're developing a new way to test the safety of our drinking water. Right now, public health officials test for contaminants like E. coli at the tap after it's already in your home. But as Linda Aylesworth explains, researchers are taking a radically different approach, one that would take a snapshot of all of the potentially dangerous organisms in the water right at the source.
Crystal clear water fresh from the mountains. It certainly looks clean, but what you don't see can make you sick, even kill you.
There's bacteria, viruses, protozoa everywhere, and they're a natural part of our ecosystem. So we want to know how they respond to changes in the environment.
It's all part of a new $3.2 million study funded by Genome BC and Genome Canada to find better ways to protect our drinking water and to alert us faster when something goes wrong.
Are these the ones that go to UBC? Okay.
One of the goals, to prevent tragedies like the one that happened in Walkerton, Ontario 12 years ago, when water tainted with E. coli killed 7 residents and made 2,500 sick.
We need better tests, we need earlier tests, faster tests, and that's what this project is about. Safer water for humans as well as animals.
Here at the BC Centre for Disease Control, water collected from taps throughout the province is routinely tested for the presence of E. coli. The process is a slow one. The bacteria are identified through a microscope and counted one at a time.
They would take the water sample, as we do here in the public health labs, and we grow it on plates, and then many hours later are able to say it's present. And what we're proposing is we need a better, faster test using nucleic acids like DNA.
This is the kind of machine that they're using to do just that. It is the latest and greatest DNA sequencer, capable of identifying not just E. coli, but pretty well everything in the water based on its DNA. It's called metagenomics.
We're looking at what we haven't even discovered. It's very exciting to know what really is present in the water.
And it can be done in a fraction of the time.
It was never possible before to analyze all the living organisms in a sample of water or in a human sample or in the soil. But now with the next generation sequencers, we're able to characterize all the things that are present in a sample.
Add to that the idea that water samples should be taken not from the tap when we're already drinking it, but from the source, and you have a promising strategy. One which, when applied globally, could dramatically reduce the 3.4 million deaths that occur annually due to water-related disease.
More sunshine in the weather forecast coming up next with Wayne.